for Chapter 13, Regulation of Gene Expression. Gene expression is sometimes referred to as the central dogma of biology. And gene expression is basically the process where the information that is held in the DNA is converted into a protein product. And this process involves two steps. The first is transcription. Transcription is where that information that is stored in the DNA is copied into an RNA molecule. And then the second step is translation, where that RNA is then used to produce the corresponding protein. So anytime we use the word gene, we are implying a sequence of DNA that holds the information to make a protein product. Prokaryotic cells must be able to regulate their gene expression. They need to be able to control when and how many proteins are produced. And this is because they live in a highly competitive environment and need to be able to respond very quickly to any changes. And they also need to be very efficient in how they use their limited amount of energy and nutrients. In order to be this efficient, they must have a very simple mechanism to control their gene expression. And the mechanism they use is called an operon. Operons are involved in transcriptional control. So remember, in the process of gene expression, DNA is copied into RNA, and then that RNA is used to make the corresponding protein. And of course, transcription is that first stage, where the DNA is copied into RNA. And so the operon is actually going to exert negative control on transcription. So negative control basically means that when you have an active molecule, so active molecule, and in an operon, that active molecule is called a repressor. So when you have that active repressor, that is going to stop gene expression. And what it's actually going to stop is transcription. So in an operon, when you have an active repressor, that is going to stop transcription. In order to understand how a prokaryotic operon functions, it's first important to know the structure of an operon. This is the diagram of an operon. And the first thing to point out is that an operon is actually a sequence of DNA. So it's a series of sequences of DNA that are involved in controlling the transcription of particular genes. So looking at the sequence of DNA, you begin with a regulatory gene. So that's the sequence that is furthest upstream, furthest to the left on the DNA. Then as you move downstream, you come across a promoter, then an operator, and then structural genes. And at the end of the structural genes, you would actually have a terminator. Now going over these sequences, some of these sequences are labeled as genes. And any sequence that is labeled as a gene means that it has a protein product. So the regulatory gene is a gene and it produces a protein product. And that protein product is called the repressor. So that's the repressor. Then as you go downstream, you go by the promoter, the operator, and then the structural genes. And these structural genes are the genes that produce the proteins that the bacterial cell wants to control the production of. And often these proteins are some type of enzyme or many enzymes. So these are enzymes. And often in an operon, to promote efficiency, all related enzymes are controlled by one promoter. So these are all enzymes that are involved in one particular metabolic process. All right, so those are the two types of genes, regulatory genes and structural genes. And remember, there is a protein product for all of these genes. Now the regulatory sequences are the promoter, the operator, and then of course the terminator. And when I talked about transcription before, I mentioned that the promoter and the terminator are signal sequences that tells the RNA polymerase where to begin transcription and where to end transcription. So during the process of transcription, the RNA polymerase will recognize the promoter, and then it will transcribe everything until it gets to the terminator. And once it gets to the terminator, it stops transcription. So pretty much the sequence between a promoter and terminator, that encompasses the genes, and that is where you find the structural genes. So the promoter is the regulatory sequence where the RNA polymerase will bind. So RNA polymerase, Pol for short. All right, then the last regulatory sequence is the operator. And the operator is where the repressor will bind. So when the repressor is bound to the operator, so this is the repressor binding to the operator, then the RNA polymerase is unable to slide along the DNA and transcribe the structural genes. So this is actually a very simple process. If the bacterial cell wants to have these enzymes expressed, the repressor needs to be removed from the operator. And if there's no repressor on the operator, then the RNA polymerase can slide along the DNA, transcribe the genes, they will get translated, and then you have these protein products, these enzymes, which can catalyze a reaction. If the bacterial does not want these enzymes produced, then the repressor will bind the operator, and that will stop the RNA polymerase from transcribing the genes. So the repressor is going to stop transcription. So if there is an active repressor, if the repressor is doing its job, it's going to stop transcription. And of course, that is negative control when that active repressor stops transcription, which leads to stopping gene expression. This is another diagram of an operon, so just going over the same information with a slightly different diagram. So this is the operon. Again, it's a sequence of DNA. And upstream, you have the regulatory gene. And when the regulatory gene is expressed, when it is transcribed into mRNA and then translated into protein, you have an active repressor. Then downstream of the regulatory gene is the promoter, the operator, and then the structural genes, and then of course a terminator. They often forget to put the terminator on. I think it's just implied. All right, so again, the operon. If you have an active repressor, the active repressor binds to the operator. So the active repressor is bound to the operator. And when that is the case, the RNA polymerase, which would recognize the promoter, the RNA polymerase is prevented from sliding along the DNA and transcribing those genes. So it's actually a very simple process. If you want to stop gene expression, you have to have an active repressor to bind to the operator, and it's literally like a roadblock. Then the RNA polymerase cannot slide along the DNA, and you do not get transcription. And then, of course, the reverse. If you want gene expression, you need to remove this active repressor. You need to inactivate it. When it's removed from the operator, then the RNA polymerase can go ahead, slide along the DNA, transcribe those genes, they get translated, and then you have the protein products. All prokaryotic operons have the same basic structure, so the same sequences that are found in the DNA. But operons can be divided into two groups, and they can be either inducible operons or repressible operons. Now, an inducible operon has the ability to be induced. So what that means is under normal circumstances, there is no gene expression. And there is no gene expression because the repressor is active, which means it is bound to the operator. but the bacteria does have the ability to induce gene expression. So it is able to induce gene expression because it has the ability to inactivate the repressor, which will remove it from the operator and allow gene expression. Now, the classic example of an inducible operon is the lac operon. And this was actually the first operon that was identified. And lac stands for lactose. And lactose, ose, is a carbohydrate, and this is a disaccharide. And lactose is also known as milk sugar, so it's found in dairy products. Now the lac operon, this is a diagram of the lac operon. The structural genes that you find in the lac operon are genes that encode the information to make enzymes to break down lactose. So they are lactose metabolizing genes. And there are about 3 genes, 3 enzymes that are involved in breaking down lactose. Now, lactose is an unusual type of sugar. So it's only produced in the mammary glands of mammals. So there isn't a lot of lactose floating around in the environment. So if the bacteria wants to be efficient and not waste time and energy, it's not going to express these genes because odds of the bacteria actually running into lactose are pretty low. So the normal situation for most bacteria is that the repressor is active, which means that that repressor is bound to the operator. So you have the repressor bound to the operator, and that prevents the RNA polymerase from binding to the promoter and sliding along the DNA and transcribing those genes into the mRNA. So that is the normal situation. Normal situation is the active repressor is bound to the operator, and that prevents transcription, prevents the RNA polymerase from sliding along the DNA. But if the bacteria do run into lactose, if they do come in contact with lactose, that rare sugar, then they will have the ability to activate those genes so they can utilize the lactose as a source of nutrients. So when that happens, the lactose itself acts as an inducer. And when we say it acts as an inducer, what the lactose actually does is it binds to the repressor. So here is the lactose, that disaccharide. It binds to the repressor, and when it does that, it inactivates the repressor. So when the lactose binds to the repressor, it's going to change the shape of the repressor, which changes its function. The repressor is a protein. And the way it changes its function is it inactivates the repressor, so then it leaves the operator, it unbinds from the operator, and now the RNA polymerase can bind to the promoter, and then it can slide along the DNA, it can transcribe those genes into the mRNA, and then that gets translated into the enzymes, and then these enzymes can break down the lactose. So this is an example of an inducible operon. An inducible operon is normally not, does not have gene expression. So those enzymes are not being expressed because there is an active repressor which is bound to the operator. But if an inducer is present, and in this case with the lac operon, it's the lactose itself, if there is an inducer, the inducer will bind to that repressor. It will inactivate the repressor, which will then allow the RNA polymerase to bind to the promoter and transcribe those structural genes. The other type of operon that a prokaryotic cell can have is a repressible operon. And a repressible operon is exactly the opposite of an inducible operon. A repressible operon is an operon where you have the ability to repress the gene expression. And the typical example of a repressible operon is the tryptophan operon. Tryptophan operon. and this has to do with tryptophan. This is the molecular structure for tryptophan, and tryptophan is a type of amino acid. And remember, the amino acids are the building blocks for proteins. And each amino acid has an alpha carbon, a central carbon, and that carbon has 4 covalent bonds. One covalent bond is to an amino group, the other covalent bond is to a carboxyl group, then a third covalent bond is to a hydrogen, and then the last covalent bond is to a functional group. And this is the particular functional group for tryptophan. But tryptophan is one of the 20 amino acids that is required by living things in order to make proteins. So tryptophan is pretty important for living cells. This is an example of the tryptophan operon. So again, the operon, it is a sequence of DNA, and it has the same structure as all the other operons. There is a regulatory gene, then downstream is the promoter, the operator, the structural genes, and then of course a terminator at the end. But for the tryptophan operon, these structural genes encode enzymes that produce tryptophan. So these are the enzymes that actually make tryptophan. And because most cells, especially bacterial cells, need a constant supply of tryptophan, the normal situation for this operon is that the repressor is inactive. So the normal situation is that the repressor is inactive, so it is not bound to the operator. So then the RNA polymerase is able to bind the promoter, slide along the DNA, transcribe all those genes. Those genes get translated into the proteins, the enzymes, and then those enzymes are producing tryptophan that the cell needs. But remember, this is a repressible operon, so you have the ability to repress that gene expression if there is too much tryptophan. So if there is too much tryptophan for whatever reason, then the cell doesn't need to waste any time or energy making even more tryptophan. That's not efficient. So if there is too much tryptophan, then the tryptophan acts as a co-repressor. So we call it a co-repressor because it works with the repressor, like co-worker or coordinate or cooperate. So the tryptophan itself is a co-repressor. So when the tryptophan binds to the repressor, it activates the repressor. So now when that tryptophan is bound to the repressor, you now have an active repressor. The active repressor binds the operator, and when it binds the operator, then that RNA polymerase cannot slide along the DNA, and it cannot transcribe those genes. So if you don't have a transcript, you don't have production of the mRNA, there's nothing to translate, no gene expression, you don't have those enzymes that would make tryptophan. So this is an example of a repressible operon. The normal situation is that the repressor is inactive, so you get gene expression. The RNA polymerase is able to transcribe those genes, they get translated, and then you have enzyme production. But if there is too much tryptophan, the tryptophan acts as the co-repressor to bind to the repressor, activate it, and stop gene expression. The inducible and repressible operons were examples of negative control of transcription. And remember, negative control of transcription is where the active molecule shuts down gene expression. So the active repressor would stop transcription, stopping down gene expression. There's also positive control of transcription, and so there are these processes that are found in bacteria. And for positive control, this involves the molecule cyclic AMP. And AMP stands for adenosine monophosphate. So this relates directly to ATP. ATP is adenosine triphosphate. When it releases a phosphate, releases its energy, then it is converted to ADP, adenosine diphosphate. And that molecule can release a phosphate, and then it's converted to AMP. And that can form sort of a cyclic structure where that phosphate binds back to the sugar. So that's cyclic AMP or CAMP. Now for positive control, this is going to involve a protein called CAP, and that stands for catabolite activator protein. And that protein is going to bind to a sequence of DNA called the CAP binding site, which is upstream of the promoter. So for positive control, it's going to involve a CAP protein binding to the CAP binding site. Well, initially this protein is inactive, but if the cyclic AMP binds to the CAP protein, that will activate it. So for the positive control, the cyclic AMP binds to the CAP protein, that activates the CAP protein, and then the CAP protein binds to the CAP binding site. And when it does that, it bends the DNA in a way that helps the RNA polymerase bind. So having an active CAP protein will actually promote transcription because it helps the RNA polymerase fully bind to the promoter, and once it's bound to the promoter, then it can transcribe the genes. Eukaryotic cells also need to be able to regulate their gene expression. And for the unicellular eukaryotic organisms, the reasons are the same as for the prokaryotes. They need to be very efficient and not waste energy or nutrients producing proteins that they don't need. And also they have to be able to respond to changes in the environment. But there are also multicellular eukaryotic organisms, and they need to control their gene regulation because they need to control which proteins are produced in the different differentiated cells. So if you remember when we talked about mitosis, referred to stem cells, and stem cells are cells that can differentiate into any kind of tissue. And the thing about the stem cells is that they have all the genes to produce all the proteins for all the different types of tissues. But when those cells differentiate, like producing an immune cell or differentiating into epithelial cells, all of those cells, like the white blood cells and the epithelial cells, they contain all the genes to make all the proteins in the human body. But immune cells need to be able to make antibodies because that's part of their job when they are protecting you from infection. And epithelial cells need to be able to produce melanin. So melanin is the protein that gives skin, hair, and eyes its color, but it also protects you from UV damage. So the immune cells do not need to produce melanin. That's not part of their job. And the epithelial cells do not need to be able to produce antibodies. So these cells need to be able to control which proteins they are producing. So eukaryotic gene regulation is much more complicated compared to prokaryotic gene regulation. Prokaryotic gene regulation is pretty simple. It basically just involves operons. But eukaryotic gene regulation is much more complicated, as there are several levels to controlling gene expression. The first level is with chromatin structure. Then there are several different controls for transcription and also translational control. And then actually they even exhibit post-translational control, so changes that occur to the protein to control whether the protein is functional or not. The first level of controlling gene expression in eukaryotic cells involves the chromosomes and how they condense. And chromosomes are a combination of DNA and proteins called histones. So that long strand of DNA is wrapped around complexes of histones, and that creates the chromatin. So that creates the molecule that we call chromatin. And then that chromatin is further condensed into euchromatin, which is fairly loosely condensed, and it's further condensed into heterochromatin. And euchromatin, because it is more loosely condensed, this is where you have active gene expression. But when it is more highly condensed into heterochromatin, that's where there is no gene expression. The chromatin remodeling complex is a protein complex that is involved in converting heterochromatin into euchromatin. So this is a complex that is going to promote gene expression. So in this example, this is some heterochromatin. So you have the DNA wrapped around the histones, so the histone complexes, and this creates nucleosomes. So that DNA wrapped around the histones. And when the DNA is wrapped tightly around those histones, when you have a lot of nucleosomes, then the promoters in the DNA are inaccessible. The RNA polymerase cannot get in and bind to them, so if you can't have the RNA polymerase binding to the promoters, there's going to be no gene expression. Well, if the cell needs to express those genes, then the chromatin remodeling complex can come in, it can identify that nucleosome and remove those histones. So it will remove those histones, and that will result in opening up that segment of DNA. The promoter will now be accessible, and the RNA polymerase can then bind to that promoter and transcribe those genes. So the chromatin remodeling complex can convert heterochromatin into euchromatin to allow gene expression. The chromatin remodeling complex can also add different molecules to the histones in the chromatin to control whether it is euchromatin or heterochromatin. And for this example, here's a nucleosome. The nucleosome has those blue histone proteins, and each blue histone protein has a tail on it. So an extension that looks kind of like a tail. And of course, here's the DNA wrapped around the nucleosome. So the chromatin remodeling complex can add either methyl groups or acetyl groups to those histone tails. And the methyl group is just a carbon with 3 hydrogens bound to it. An acetyl group is a carbon that is bound to an oxygen and then also bound to a methyl group. If methyl groups are bound to the tails of the histones, then that will result in the formation of heterochromatin, which is highly condensed, and there will be no gene expression. If instead acetyl groups are bound to those tails for the histones, then that creates euchromatin, which is more relaxed, not quite as condensed, and you can get gene expression with the euchromatin. And the chromatin remodeling complex can control whether the methyl group or the acetyl group are added to those histones. So it can control whether that segment of DNA is found in heterochromatin or euchromatin. But the methylation of the histones will lead to less or no gene expression. DNA can also be methylated. So for DNA to be methylated, that involves the cytosines having methyl groups attached to them. So that's the cytosine with a methyl group attached to it. And when DNA is directly methylated, when the cytosines have the methyl groups added to it, this is a more permanent type of inactivation. So when DNA is methylated, then these genes that are in the segment are unable to be expressed. And this is a more permanent type of inactivation. Whereas methylating the histones, this is reversible. One example of how DNA methylation permanently inactivates a chromosome is with the bar bodies. And bar bodies relate to cells that have 2X chromosomes. So those are the examples of the X chromosomes. And these are seen in females. And remember that these are homologous chromosomes. So one chromosome came from the mother, maternal chromosome. The other came from the father, so that's the paternal chromosome. So as the original zygote which received these two chromosomes, as it divides and forms daughter cells, it's going to be random which X chromosome is inactivated in the daughter cells. So in this daughter cell, the paternal X chromosome was inactivated. In this daughter cell, the maternal X chromosome was inactivated. And when we say inactivated, it means that DNA is methylated and highly condensed. So you see this little dark staining area called a bar body in the cell. And in this example, this is a classic example of X inactivation in cats. And on the X chromosome, there are the genes for fur color. So the maternal chromosome had the gene for orange color. The paternal chromosome had the gene for black color, fur color. And so what you see in the cat are calico cats. So calico cats, where you see those random spots of orange fur, random spots of dark fur, that's all due to X inactivation. And that's because it's just random. The cells in this area inactivated the maternal X in this example, and the cells in this area inactivated the paternal X chromosome. And so all calico cats have to be XX. So all calico cats are female. There are a couple exceptions. There are a few male cats that can become calico, but then they have to have two X chromosomes and a Y. So they have to have that X inactivation. That is what leads to the differences in color. Eukaryotic cells can also control whether transcription occurs or not. And remember, transcription is where the DNA gets copied, the information gets copied into the RNA. And eukaryotic transcriptional control is very complicated. So it can involve activators and/or repressors, and it involves many protein-to-protein interactions. This is one example of eukaryotic transcriptional control. And in this case, you have the molecule of DNA. So this is the strand of DNA. And there is a sequence of DNA called the enhancer. So the enhancer is going to promote gene expression. And the enhancer is usually pretty far upstream from the promoter. And remember, the promoter is where the RNA polymerase binds. And it's right before the gene. So the RNA polymerase will bind here, and then it will slide along the gene, transcribing the gene, and then that mRNA gets translated and you have gene expression. Well, in this example, there needs to be a transcription factor complex that will help the RNA polymerase bind to the promoter. So the RNA polymerase cannot bind by itself to the promoter. It needs this complex of proteins to help it do so. And in this example, a transcription activator will bind to the enhancer. And once you have that transcription activator, that protein bound to the enhancer, then that causes the DNA to bend. And now that enhancer lines up with the promoter, and between the two you have a lot of proteins binding together to form the transcription complex. So that will cause this DNA to fold over, line up the enhancer with the promoter, and these proteins are all bound together. And having that transcription complex, that will allow the RNA polymerase to now finally bind to the promoter and transcribe the genes. So this is one example of transcriptional control in a eukaryotic cell. And it involves a sequence of DNA called an enhancer, and then many types of proteins, mediator proteins, transcription factor complexes to form in order to help or assist the RNA polymerase to bind and then allow it to transcribe the genes. After transcription, the eukaryotic cell can continue to control gene expression by alternate splicing of that mRNA. And I've mentioned alternate splicing before. Splicing involves removing the introns from the pre-mRNA and leaving the exons to be translated. So remember, intron relates to interrupting sequences and exon is express sequences. Well, alternate splicing allows for different exons to be left in the RNA so that you get different proteins that are produced. So in this example, the exons are A, B, C, D, and E. And after some splicing, you've removed all the interrupting sequences, the introns, and so you have the complete protein, A, B, C, D, E. All of those segments will be translated, and this is your protein product 1. Well, if, for example, this type of protein is not required, the cell needs a modified protein, there can be alternate splicing that occurs. And so you start with the same pre-RNA, which has the segments A, B, C, D, and E. And in this case, C is going to be spliced out. So the resulting mature RNA is A, B, D, and E. So you've spliced out C, and now the resulting protein is a different protein product. So alternate RNA splicing will allow the cell to produce slightly different proteins when needed. Another example of post-transcriptional control in eukaryotic cells is RNA interference. And RNA interference involves small segments of RNA preventing translation. So an example of this is that DNA will be transcribed into an RNA molecule, and then that RNA molecule will be broken down to form these small RNA segments, and these are the small RNAs. And then those small RNAs can inhibit translation in 2 different ways. Some of these small RNAs will be miRNAs, and that stands for microRNAs. And those microRNAs are going to bind to the mRNA. So this is the mRNA that was produced by translation of the gene. And these miRNAs are going to bind to the mRNA. And when they do that, then when the RNA leaves the nucleus and goes to interact with the ribosome, it's going to stop translation. So it's going to prevent the ribosome from translating that mRNA. The other way these small RNAs can prevent translation is by siRNAs. siRNAs are called small interfering RNAs, and they will bind together to form a complex called RISC. And that RISC molecule is going to bind to the mRNA and degrade the mRNA. So it's going to break down that mRNA into little pieces. And of course, if the mRNA is degraded, then you're not going to have any translation. So RNA interference involves small segments of RNA binding to the messenger RNA and preventing translation. RNA processing can control gene expression at the transcriptional level and also the translational level. So at the transcriptional level, the alternate splicing can control what type of protein is produced. In terms of translational control, the RNA processing involves that cap and the poly A tail. So the 5' cap, that guanine that is added at the 5' end of the mRNA, this is going to determine if the ribosome can actually bind to the RNA. So not having the 5' cap will stop translation. The poly A tail, adding adenines to the 3' end of the mRNA, that's going to determine how many times this RNA can be translated. So the longer the tail, the more proteins will be produced from this mRNA. So RNA processing can involve cap, putting on the 5' cap, splicing out the introns, and adding the poly A tail. So splicing will help control at a transcriptional level. Adding the cap and the poly A tail will help control translation. The last point at which a cell can control gene expression is after the protein has been made. And this is called post-translational control. So translation, of course, is the process by which the mRNA is used to make the corresponding protein. So once the protein has been made, the only way to control that enzyme is by allowing it to remain or to degrade it, to break it down. And so there are certain molecules called proteases that break down proteins. So when a cell wants to get rid of a protein, it is going to tag that protein. So there are certain molecules that will bind to the protein and it will tag that protein. And once the protein is tagged, then that protein will enter a proteasome. And the proteasome is the complex that is going to break down the protein. So a tagged protein will enter the proteasome, and then the proteasome will break down that protein into its various amino acids. And of course, if a protein is broken down, then it is no longer functional. The last topic that I want to talk about in this lecture is mutation. And a mutation is defined as a permanent change in the DNA of an organism. And the change in the DNA may or may not affect the resulting protein. Now, the effects of mutations can be different for different types of organisms. For unicellular organisms, often a mutation that affects the protein can have a lethal effect, so it can cause the death of the organism. For multicellular organisms, there can be different effects. For multicellular organisms like humans, having a mutation in a somatic cell, if it affects the protein, it can lead to cancer. But those mutations are not transmitted to offspring. Only mutations that occur in gametes, which it's pretty rare, but if the mutation occurs in the gamete, that will be passed down to the next generation. Now, the causes of mutation. There are two basic causes of mutation. The first cause of mutation is called a spontaneous mutation. And a spontaneous mutation is when the cell makes a mistake. So often this is during DNA replication. So this is an example of DNA replication. Here is the original parent molecule. This is the replication fork. And both original strands are being replicated. And it's possible during this process of DNA replication that the proteins, the DNA polymerase, makes an error. But this is very, very rare. This is a very rare event, but it's called a spontaneous mutation. Much more common are induced mutations. And an induced mutation is where the environment causes a change in the DNA. And you are probably pretty familiar with most of these different types of things. These are mutagens. So these are different things that can mutate the DNA, causing a permanent change in the DNA. So first is radiation. UV light, you probably know that exposure to too much UV light can lead to skin cancer. X-rays, gamma rays, they have even more energy than UV, so that can cause more damage and lead to cancer. There are different types of chemicals that you can find, especially in cigarettes or in food or other chemicals that people use, herbicides, pesticides that can alter the DNA. And there are even microbes that can alter the DNA, especially viruses. We're finding more and more viruses are associated with cancer. HPV is often associated with cervical cancer. And there are even some bacteria, like Helicobacter, that can lead to stomach cancer. So a mutation is defined as a permanent change in the DNA, and mutations can be caused by spontaneous mutations, and that involves normal processes in the cell, or they can be induced by mutagens. And so those mutagens are things from outside the cell that damage the DNA. Now I want to go through a couple of different types of mutations and what effect that can have on the cell. So in this figure, this is the normal situation. So it shows you the normal strand of DNA. This is the template strand, and this is the complementary strand, and this is the normal sequence. And this would be the normal mRNA that is transcribed from that DNA, and this is the normal protein that is encoded by that sequence. All right, so this is the wild type. Wild type is the term we use when we're talking about the normal sequence of DNA and the normal protein. For this shows a point mutation. So a point mutation is when only one base pair is affected. So this is the base pair, so the base and its complementary pair in the other strand of DNA. So that's what we refer to as the base pair. So originally in the wild type, this is the original base pair, and this is the mutation. So when you have this mutation, this is going to cause a change in the RNA. So that's going to change the RNA. And then when you actually go through and translate this, though, you end up with the same exact protein. So the last amino acid in the wild type protein was glycine, and it is still a glycine. So we refer to this as a silent mutation. So there is a mutation, the DNA has been altered, but the resulting protein product is unaffected, so it's a silent mutation. So there should be no effect on the cell. So the cell should be able to function the normal way. This slide shows another point mutation that has a different result. So again, this is the same wild type example. This is the DNA, same sequence of DNA, same sequence of RNA, and the same protein that's produced. Now in this case, you have a different point mutation. So now this is the base pair that has been altered. So now this was the normal wild type base pair, and now it has been mutated. And with this mutated, once you go through translation, it actually creates a change in the amino acid sequence. So originally, where the original protein ended with glycine, now it ends with a serine. And this is called a missense mutation. So one of the amino acids in the protein has now been altered. Now the effect this would have on the organism is unknown. So if this is a really important amino acid for that three-dimensional structure of the protein, and if this change leads to a change in the structure of the protein, then that will probably have a negative effect on the cell. Because the assumption you want to make is that every protein that a cell produces is important to the function of the cell. So if it produces a protein that can no longer function, that's probably going to have a negative effect on the cell. But it's possible that this amino acid is not important to the shape of the protein, and so it is possible that this protein could still remain functional. And in that case, it would be considered a silent mutation. But just a missense, unless you see exactly how the protein functions, you don't know if this is a silent mutation or a lethal. Lethal implies that the cell cannot survive without the normal protein. This is another example of a point mutation. So again, this is the normal situation. And now this base pair has been mutated. So instead of a thymine, it was altered to an adenine. So that's the change in that base pair. And now this leads to converting this codon into a stop codon. And this is considered a nonsense mutation. So a nonsense mutation is when you have an early stop. And this is probably going to be a lethal mutation, because what you notice is now you no longer have that normal protein. So the normal protein was this, methionine, lysine, phenylalanine, and glycine, and now you don't have that protein. And again, the assumption is every protein that a cell produces is important for that cell's function. So if that protein is no longer present, then that's going to have a very negative effect on the cell. And we refer to that as a lethal mutation. The last type of mutation I want to talk about is a different type of mutation, and this is a mutation where there is a change in the number of nucleotides. So again, the normal wild type situation, this is the wild type DNA, the wild type mRNA, and the wild type protein. So wild type protein, methionine, lysine, phenylalanine, and glycine. So now in this mutation, now there's a deletion. So this adenine and thymine are going to be deleted. So going to be deleted, so that's going to cause a change in the mRNA. So now the mRNA is missing that uracil, and what that will cause is that at that mutation point, that will cause a change in all the following amino acids. And this is referred to as a frame shift, because it's going to cause a change in the reading frame, how you read those codons. It's going to be shift, and the result is going to be extensive missense. So a missense is a changed amino acid, but you're going to have a lot of different amino acids. So that's the extensive missense. And this is going to create a completely different protein. So if you compare the wild type protein to the new protein, they are going to be totally different. And often this will be a lethal because you no longer have the original protein to perform its important function for the cell. Now I want to go through a couple practice problems involving gene expression and mutation. So the first question, what would happen to the protein product if the third C was mutated to a T? So first thing you want to notice is that the sequence involves DNA. It involves DNA because you see thymine in it. So this is a template strand of DNA. And now it's asking you what would happen to the protein product if there was this particular mutation. So in order to understand what's happening to the protein product, you have to know what is the normal protein product. So the very first thing you need to do is take the normal sequence of DNA, transcribe it, and then translate it. So the transcription here, if I were to do it in green, it's complementary base pairing into RNA. So know that G will complementary base pair to cytosine, adenine to uracil, cytosine to guanine, thymine to adenine, adenine to uracil, cytosine to guanine, and so on. And what I've done is just added the actual sequence, so I don't have to write it all. But that's the first thing you want to do is figure out the mRNA. And once you have figured out the mRNA, then you need to translate it. So to translate it, you need the genetic code, and you need to identify your reading frame. So the reading frame always starts with the start codon. So you need to slide along the mRNA and identify the start codon, which is AUG. So you slide along, and here is the first AUG. So that is going to set your reading frame. So every 3 nucleotides is a codon. So you just divide it into codons until, of course, you reach the end where you can't divide that into a codon. Then the next thing you do is you translate. So you look up the codon and write down the corresponding amino acid. So AUG, it's methionine, so that would be met. And then the next one is UCG, so the U's are here, C here. UCG is serine, so that would be serine, that's the next one. Then AGG, so A is here, G is here, so you go down to AGG. That's arginine. Arginine, so attach that together, peptide bond. CGU, CGU, CGU is arginine also. Arginine. And then UAA, UAA is stop. So when you get to the stop, you stop. So the protein product is methionine, serine, arginine, arginine. That's the normal protein product. So now you want to see what will happen if the third C was mutated to a T. So third C 1, 2, 3. So this is now mutated to a T. So when doing that, the third C, let's see, this is AUG here. So the C would be mutated to a T, so that would change that to an A. So that nucleotide in the RNA would be changed to an A. So now this codon is UCA. So UCA is still serine. So there would be no change in the amino acid. So this is a silent mutation. Silent mutation. For the second practice problem, I'm using the same sequence of DNA, but a different mutation. So again, remembering that this was DNA and that it had to be transcribed into RNA, and then this has to be translated. So again, for translation, you want to find the start codon. This is the start codon, and I'll just correlate it with the DNA to keep everything even here so that it's easier to see what's going to be affected with the mutation. So it's pretty close. All right, again, translation, just going through translation, AUG. AUG is methionine. And then UCG. UCG is serine. And then the AGG. AGG is arginine. And then the CGU. CGU is arginine. And UAA was the stop. All right, so that is your original protein, your functional protein. Now this mutation is if the third A was mutated to a C. So third A, 1, 2, 3. And if this was mutated to a C. So now it's no longer an A, it's a C. So this would not be a U, it would be a G. So now this codon is GCG. So GCG is an alanine. So that would convert this serine to an alanine. And this would be a missense. Because now you have had a change in the amino acid. And the effect on the cell could be a lethal, it could be a silent mutation. You don't know until you can see how this new protein functions. This is the last practice problem with the same sequence, and it's a different mutation. So again, this is your DNA, this is the transcription, and again, you have to find the AUG, the start codon, and that sets your reading frame. So just correlating the DNA with the RNA to make it a little bit easier to follow where the mutation would be, and that one. And translation, when you use your genetic code to translate it, this is methionine, and then serine, arginine, and this again was a stop. All right, so that's your original protein. So now this mutation, this is a change in the number of nucleotides, or the change in the number of base pairs. So now this mutation is asking what would happen if the second G was missing. Second G was missing. This is the first G. This is the second G. So now this G is going to be missing. And this is going to change the RNA. So it's going to change the RNA because now you no longer have the C. So this is going to change the reading frame. And now the start codon won't change, it's AUG, but now the next codon is UGA. Then it's GGC, then GUU, then AAG. So this is going to change the protein. So AUG will still encode for methionine, but now you have UGA, and UGA is a stop. So that's a stop. So this is going to create a nonsense mutation. A nonsense is an early stop. So now this protein has been eliminated. And the most likely result of this type of mutation is a lethal. I mentioned before that one possible result of mutation in a multicellular organism like a human is cancer. And that's where one individual cell has a mutation that affects control of the cell cycle, and that causes that cell to go through mitosis uncontrollably, and that leads to a tumor. So a mass of cells that interfere with the function of the normal tissue. And there are many different ways to acquire mutations that could lead to cancer. There's heredity. You can inherit a mutation from one of your parents. You can be exposed to radiation or exposed to chemicals, or again, even viruses. We are finding a lot of viruses are actually associated with triggering cancer. And the two most common genes that can be mutated that will lead to cancer are proto-oncogenes. And proto-oncogenes are genes that promote mitosis. They promote the cell to divide. And the other type of genes are tumor suppressor genes, and that stops cell division. So if a tumor suppressor gene is mutated, then it can no longer stop cell division, and that also leads to uncontrolled cell growth. This is the end of the lecture for Chapter 13, Regulation of Gene Expression. And gene expression involves taking that information that's in the DNA, making an RNA copy, and using that RNA to produce the corresponding protein. And for cells, it's very important to be able to control which proteins they are producing, because it helps them to be efficient, to adapt to changing environments, and for eukaryotic cells to allow the different types of cells to produce the different proteins that they need.